So this is my model, and sorry for turning it in very late. So here you can see the model separately with all the vocab, and this is the double-stranded DNA. Then this is the final model piece. So transcription and translation are processes used to replicate RNA strands from DNA to use to code for proteins that are essential to the function of the human body. The process starts with transcription, which is the process of conversion of DNA into RNA. This is the double-stranded DNA used to start off the process. It's a double helix structure composed of a phosphate group and a sugar group, as well as 4 nitrogenous bases called nucleotides, which are specifically, there are 4 of them, and they're known as guanine, cytosine, adenine, and thymine. So let's start off with transcription first. Transcription, the RNA polymerase uses DNA strands as templates to make new complementary RNA molecules during transcription. The splitting of DNA strands is specifically done by the helicase, which unzips the DNA to expose the nitrogenous bases within. At the very beginning of this process, a 5 cap is added to the first nucleotide and the transcript. This cap is a modified guanine nucleotide, which protects the transcript from being broken down. The process also uses transcription factors, and these are basically the proteins which involve the process of converting and transcribing DNA into RNA. Throughout the entirety of transcription, single-stranded binding protein bind regions of DNA during DNA replication and recombination, as well as DNA repair. The poly A tail makes the RNA regions of DNA. Where'd it go? Yeah, the poly A tail makes the RNA molecule more stable to prevent the degradation of both transcription and translation. And that's the entire model for transcription right there. And that's the DNA. And now we'll be talking about translation. So translation is the next step after transcription and manufacturing proteins in the body. Small ribosomal units, as seen here, they bind and read the mRNA during translation, and mRNA nucleotides and codons then make the proteins, as seen here. An mRNA of a specific structure makes the proteins of a specific structure with the help of tRNAs, right here, with the help of tRNAs, which help decode a mRNA sequence into that protein. The large ribosomal subunits, right here, take care of production. It contains the peptide transfer site, the site at which peptide bonds are basically formed. And after the translation process is finished, various amino acids are produced, which are the products of the transcription and translation of RNA into the protein it specifically codes for. So here you can see the small ribosomal subunit with the mRNA strand at the top, and these are units of tRNA at the top, and they help code for the specific codons help code for the specific proteins. And here are the large ribosomal units, which contain the peptide transferase site and the site at which peptide bonds, as seen here, are basically formed. Over here on top, we see the transcription factors and the non-coding DNA, as well as the template DNA strand, which is basically replicated throughout the process of transcription, as well as the helicase, which unzips the DNA to allow the formation of unveil the nucleotides, which are hidden within.